Hi.
Hello. Are there any questions? Thanks for confirming whoever said something. Well, it's rather big. Shrink that down a bit. All right, today is May 3rd. Let me announce some things before show you my screen. And that is, I did a lot of grading this weekend. I got 2 of the earlier labs done. I think Lab 00 and Lab 01. They are now finalized. I've gotten last week's lab graded the first time. Let me see now, what lab was that? Lab 5 is graded for the first time. And then I'm starting to grade Lab 6. I'm about halfway through the class on Lab 6. I'll hopefully have Lab 6 finished tonight. So for Lab 5 and Lab 6, well, I should say for Lab 5, and if I've graded your Lab 6, get the updates to the grades in by this Saturday, and then you will get up to full credit.
Hey, so I have a question. So I was after my lab got graded, I was trying to resubmit for the second time, but it doesn't have the window open to submit it for the second time for lab 5 and 6.
It
It
Lab
says it was
5, you can't submit. Let me check on that. It should be, because
says it
should
was
be by the,
lost.
just a second. It should be until the until date. It will tell you you're submitting it late, but you can ignore that as long as you can submit it. There isn't a second due date in Canvas, and I have 2 due dates. So Lab 5, it says here, let me show you this. Just a minute, I have to remember how to show the screen. Come back to this window. Open that up. Share the screen. So this is worksheet Lab 5, and it says you can submit it until April 30th, which that isn't right, is that? Oh. Yeah, for some reason that one had the due date the same as the until date. The until date should be a week less, later than the due date. And let's see, the 30th is Saturday. I'm confused here. Can't be the second. Why was that April? Oh, that's March. That's why. April 30th was Saturday. So it's the 7th. There we go. Sorry, I just got confused on the month there. All right, so Lab 5 you can turn in. Lab 6, I will get that straightened out later. Someone can remind me in the lab or after the class is done with. I don't want to get in the time now. Let me stop sharing my screen, and then I will tell you a little bit more about what I got graded. I'm showing the grade book, which is why I do not want to show you the screen. So Lab 5 is graded. Lab 6, I have about half the class graded. Lab 0 and Lab 01. And then the last lecture worksheet, I don't remember what that was. Anyone remember?
Lecture 4.
Is it lecture 4B? Yeah, I got that graded. Oh, and I got all of the extra credit study groups graded too. I need to go back and grade. It's one of the quizzes. It's an essay quiz. Look at the quizzes here. I was hoping to get to at least one of the quizzes this weekend. Somebody asked a question. There is an extra credit question. I think it's quiz 2B. I've not gone in and given you the extra credit to quiz 2B yet, so I need to do that. And oh gosh, I haven't done quiz 1A or quiz 2A. So I was hoping to get to those this weekend. I did not get to them. All right, any questions about the grading?
Quick question. So tonight we're getting tested on back of chapter 3, half of chapter 3 and beginning of chapter 4. I'm a little confused what is getting.
It's in the syllabus. I'll show you that in a minute here.
Thank you.
Let me go to worksheet lab 6A, and then I'll just open the window. I'm not gonna do that. All right, I'm shutting down the grade book, so now I can show you screen. So you should be seeing the screen. Go ahead and edit there too. So what today is May 3rd, and we're covering chapter 5. I think we need to finish chapter 4. I don't think we finished chapter 4, and I need to talk about the symbiotic theory at the very least. So anyways, there's a question about quiz 3 on the syllabus. It always states what it is. It's covering the end of chapter 3 and chapter 4, and I haven't finished chapter 4 yet, so let me finish that quickly. And then it also covered lab 5 and lab 6 that we did last week.
I have a question on that. Our lab 6 is not graded yet. How do we know?
I will try and get lab 6 graded the first time tonight. Okay.
I just need to take my test after the Zoom class because I don't have any other time besides this.
Well, that's a bad situation there then. Speak to me in the lab and I'll try and get your lab 6 graded first among all the other labs. All
Thank you
right?
so much. Thank you.
All right. But remind me because I won't remember this.
Yes, I will.
Too many things going on, so you got to remind me. All right. Let's see. And then we're having a lab today, lab 7. And I need to discuss the start of the unknown project. And I think what I decided to do last week is we're only going to discuss the unknown project. And so there is no lab 7. We'll cover that on Thursday. All right. Any questions about what we're doing? If not, let me get to chapter 4. And I think we were talking about the eukaryotic cell structures. And I did not finish that. I'm going to assign that to you guys because this should be review for what you had in Biology 160. Well, hopefully I got further than here. Let's see if I made another one. Yeah, I did. So we were talking about passive transport and osmosis. You know, I got further than here even. I remember talking about this. All right, so we talked about endospores. So that's great because the next slide is talking about eukaryotic cell structure. And there, I got 2 here all over in this lesson. I need to remove those 2 here's when I'm done with them. And you saw how many I had. Okay. In fact, I seem to recall I talked about vesicular transport. All right, great. So learn about the cytoplasm, the cytoskeleton. I'm just going to show you, this is a picture of a cell and the cell membrane. You can't see it, but it's someplace over here. And there's another picture of a cell. What we're looking at is the cytoskeleton, not the cell itself. So that's why you're not seeing the edge of the cell here. And we're not seeing it here, but it looks like we're seeing the entire cell there. And that's the backbone of the proteins running through the cell that make up the cytoskeleton. And it's very extensive, the cytoskeleton in eukaryotes. Let me mention just one other thing. And that is there are vacuoles in animal cells. And there are vacuoles in plant cells, but there's only a central vacuole in a plant cell. The central vacuole is the largest organelle in a plant cell. It's larger than the nucleus. In an animal cell, the largest organelle is the nucleus. And let me briefly mention the eukaryotic endomembrane system is a system for moving mainly proteins about in the cell. And it involves the endoplasmic reticulum and the ribosomes make proteins. The rough ribosomes make proteins. Where the heck is, there it is, the rough ER, make proteins. And then they will package those proteins in a vesicle and ship the vesicle to the Golgi apparatus. The vesicle will fuse, right there showing the vesicle fusing with the Golgi apparatus. And then the proteins will be modified in the Golgi apparatus. And then they will be put into a vesicle, which will butt off the Golgi apparatus. And then that vesicle will move to where the protein should be. It could be another organelle. The vesicle could fuse with the cell membrane and then dump the proteins outside the cell. Here we go. The endosymbiotic theory. The endosymbiotic theory is a theory to explain the evolution of mitochondria and chloroplasts in eukaryotes. Go ahead and make a new window. I probably should use paint though, because then I can draw. So let's skip that. Sorry, I've got too many windows open here. I don't think I can close that one. The theory of the endosymbiotic theory, which is the endosymbiotic theory, is that a long time ago, there was a proto-eukaryote cell, meaning a cell before eukaryote cell, and it was a large cell. And it engulfed a small cell, which I'm just going to draw it like this, which I'm not having any luck with my mouse to finish. There we go. And it got engulfed by the proto-eukaryotes. So let's go P here for proto, and then E for eukaryote. This small cell here was actually a bacteria, or we can call it a prokaryote, but it was a bacteria. And it engulfed that small cell. It may have engulfed it by phagocytosis, or it may have engulfed it by pinocytosis. We're not entirely sure. So eventually we're going to erase this now because it's going to move into the cell. And then the little cell, trying to erase that, there we go, was incorporated in a vesicle inside the bigger cell. And for whatever reason, the big cell didn't eat the little cell right away. Any questions about that part? All right. Well, because life was early back then, there were some things that happened, like there was glucose in the cytoplasm of the proto-eukaryote cell, and it diffused into the little cell. Well, the big cell could only perform fermentation to derive ATP. And I'm sorry, this is so small. Let's see if I can blow this up. That'll be better. So ATP, glucose, can you spell that right? Could be made into ATP, but using fermentation, if you remember, you only get 2 ATP. Any question about that? Well, the little cell, which is the bacteria, could perform aerobic respiration. And so it took that glucose and it made, well, probably about 38 ATP. And we don't know how many of those ATPs left. We draw again. Left and went into the cytoplasm of the big cell. But like I said, life was early. And so when, in a sense, a mistake was made, meaning the little cell made ATP and some of it diffused into the big cell. And it was much more than 2 ATP diffusing into the big cell. And we don't know what it was, but it was someplace between 2 to 38 ATP. I'm not getting that to right. Sorry. Put it right here. 2 to 38 ATP moved into the big cell. Well, the big cell said, gosh, I lost 1 glucose. I gained a whole bunch of ATP. This is good for me. I can't make that much ATP from 1 glucose. So I'm going to keep this little guy around and he will make me ATP. And so the two started living in symbiosis. The little cell got a home and it got fed glucose. I'll try and draw that in here. Sort of an arrow. It's hard doing it here. And then we'll put this here, glucose. So it got a home and it got fed. And then the little cell made the big cell ATP. And so they started living in symbiosis. They've lived in symbiosis now for so long, we don't find the little cell anywhere except inside the big cell. And essentially all the big cells, there are a few exceptions. I think there are some amoeba that are big cells, which don't have the little cell in it, but essentially all the other big cells, which are all the eukaryote cells, except for a few amoebas, have the little cell in it. And so they lived in symbiosis now for a long time. Any question about any of that? Anyways, that's the theory that the big cell, which is a proto-eukaryote, incorporated a little bacteria cell, which then evolved into becoming the mitochondria within the little, the big cell. And now we can call this a eukaryote cell, no longer a proto-eukaryote. Any question about any of that? Oops, I can't erase that. Why is that? Well, see if I can erase it this way. Oh, I can. Okay, well, let's look at some evidence for the theory that a proto-eukaryote incorporated a cell, which then lived in symbiosis and eventually evolved into the mitochondria of the big cell. Well, the first evidence for this theory is that the mitochondria has two membranes. It has an inner membrane and an outer membrane. And you would expect two membranes, one being the membrane of the original bacteria, and then one being the membrane that was the vesicle surrounding the bacteria. And mitochondria have two membranes. The second piece of evidence is that we can find in the amoebas and then some other protists that they will incorporate another cell and then use that cell for the benefit of the big cell. And it'll benefit the little cell because the little cell is given a home and it's usually given food as well. In fact, the amoebas that lack a mitochondria, what they have to do is they find a little cell and then bring it in, and it's a bacteria. And then that bacteria performs aerobic respiration for the amoeba. So that there are actually some amoebas that seem to be doing this today. And they're not swallowing a bacteria that we call exactly a mitochondria, but it's a similar bacteria. And here is a paramecium that is incorporated in algae or a photosynthetic bacteria. And then the paramecium gives the home for the photosynthetic cell and then may give some nutrients to the photosynthetic cell. And then the photosynthetic cell takes light and CO2 and then makes glucose. And it shares some of that glucose with the paramecium. So we do see this going on today. And then some other evidence for some symbiotic theory is when we look at the DNA of eukaryotic cells, let me blow this up a little, the DNA is linear, okay? When we look at the DNA of prokaryotic cells, the DNA is, it's usually one circular chromosome, okay? Usually it's one circular chromosome. When we look at the DNA of mitochondria, it's one circular chromosome. When we look at the histones in the eukaryotic DNA, they're present. They are absent in bacteria, and they're absent in mitochondrial DNA. When we look at the first amino acid made in protein synthesis, you'll recall that I told you for eukaryotes, it's methionine. So the start codon always codes for methionine in eukaryotic cells. Now, I didn't tell you, but in prokaryote cells, the first amino acid synthesized is formylmethionine. It's a similar amino acid, but it is a different amino acid. And we look at the first amino acid made in a mitochondria, it's formylmethionine. When we look at the ribosomes in eukaryotes, they're 80S in size. The prokaryotes are 70S in size. The mitochondria has a 70S ribosome. And this is a gene on the mitochondrial DNA, by the way. When we look at how cells divide in eukaryotic cells, they divide by mitosis. Prokaryote cells divide by binary fission. The mitochondria divides by binary fission, and its replication is independent of the replication of the eukaryotic cell that the mitochondria is in. And then the strongest evidence for the fact that mitochondria at one time were a bacteria, when we look at the DNA sequence of the mitochondria, the sequence is very similar to the bacteria. So that if you were to say which domain the mitochondria falls into, by the sequence of DNA, it falls in the domain bacteria. Any question about that? And when did I talk about domains? I don't think it was this lesson, but if you go and look at that.
Will this slide be available in the modules?
It'll be available to you as soon as I finish the lesson. And I just finished the lesson now. Okay. All right, any questions about the theory of symbiosis to explain how a eukaryote cell obtained a mitochondria? And then in plant cells, this happened twice. A bacteria was first brought into the plant cell to become the mitochondria. And then another bacteria was brought into the plant cell to become the chloroplast. And so the chloroplast is a photosynthetic bacteria that was incorporated into a plant cell. Any question about any of that? And then the same evidence that I told you for the mitochondria applies to the chloroplast, all of the evidence. All right, that's the theory of endosymbiosis, and that is that mitochondria and chloroplast were at one time bacteria, and they became incorporated in a proto-eukaryote cell. And that actually changed the proto-eukaryote cell into the eukaryote cell. And that's how these organelles originated. And they're the only organelles in a eukaryote cell that has DNA in it. I forgot to mention that. The chloroplast and the mitochondria. All right, any questions about the endosymbiotic theory? No, let me close that. And while I'm thinking about it, I got it up here. I don't have it up here, so remind me in the lab and I'll make that slide available to you. Or you can remind me at the end of the lecture. Because I gotta go there, which I can go there soon enough. But. All right, any questions about, was that chapter 4? I now need to talk about mitochondria metabolism. I don't think I did that. Oh, I'm not wearing my headphone. That's why I was able to plug that in. So let me put on my headphone and I got to transfer this over because I don't think I have lesson 5 here. Yep. All right, any questions? Yeah, I forgot to.
I have a question about the unknowns project, but I don't know if, do you want me to ask it now or in the lab?
You should wait for the lab for that one.
Okay.
Okay, I'm going to be talking about the unknown project today, aren't I?
So are we in chapter 4 or are we in chapter 5 and 4?
We're just about to start chapter 5.
Okay.
We're now done with chapter 4.
Okay.
Remove that, yes, okay, I can pull that out. If I don't have the headphones on, you notice that, just remind me. I totally forgot today. There, can you guys hear me?
Yeah.
Okay, great. All right, so as is normal, I give you a slide which gives you the major goals and a rough outline. Of course, know the terms of the chapter, and the terms are fairly important in this chapter. We're going to be talking about microbial metabolism. And of course, the other important part of this is to know about the metabolism. We'll be specifically talking about the metabolism of bacteria and prokaryotes, but it does apply a lot of it to the metabolism of eukaryotes. And then understand what feedback inhibition is, and understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. Any question about any of this? All right. When we're talking about metabolism, it is all of the chemical reactions occurring within a cell. And you could find that as all of the chemical reactions within an organism. But as you know, with the bacteria, the organism is the cell. Metabolism can be broken down into two parts, catabolism and anabolism. Catabolism is a decomposition reaction where a large complex molecule is broken down into a simpler molecule, such as when starch is broken down into glucose. The breaking down of a large molecule to small molecules will release energy. So these are energy releasing reactions. Now, whenever energy converts from one form to another, we will always lose some of that energy as heat. And heat is a form of energy. It's just a different form of energy that isn't very useful to cells, although warm-blooded organisms can use heat to help. They can use heat to help them keep warm, meaning maintain their body heat. But other than that, heat is pretty much wasted because it can't be used for anything useful like doing work within the cell. Some of that energy that is converted from a complex molecule to a smaller molecule, when that energy is released, sometimes that energy can be used to generate ATP. Now, let me warn you that our cells, and most eukaryote cells, but our cells in particular, meaning animal cells, are really lousy at generating ATP. We only have 2 chemical reactions to generate ATP. And that is aerobic respiration. We'll talk about that process. And then fermentation, which actually that's a process too. It's not one chemical reaction, but it's a process. And those are the only 2 ways our cells can generate ATP. All of the other catabolic reactions in our cells do not generate ATP. And that is by far the vast majority of catabolic reactions. Even in cells which are much better at generating ATP, like a photosynthetic plant cell or a photosynthetic bacteria, there's only a few reactions that can generate ATP. So don't get confused by this slide. Most catabolic reactions do not generate ATP. For example, when you were to split two amino acids hooked together into two monomers, you cannot generate ATP. Another example would be when you split sucrose into glucose and fructose, you cannot generate ATP. Any question about any of that? All right, the other division of metabolism that's not catabolism is anabolism. And these are biosynthesis reaction, where smaller molecules are put together to form a larger complex molecule, such as when glucose is put together to form starch, or when amino acids are put together to form proteins. These reactions take energy, so they use energy. They're energy using anabolic reactions. And so for the reaction to occur, you have to add energy into the system. Now, whenever you transfer energy from one form, the simple molecules, to the large molecules, you always lose some energy as heat. Okay? Excuse me. Not sure where that came from. That energy that you use to convert the simple molecules into the large molecule usually comes from ATP. So ATP will be broken down into ADP and inorganic phosphate. And then the energy coming off of the ATP can be used to add on the simple molecule onto the larger molecule. Any question about any of this slide? All right. I need to talk briefly about a metabolic pathway. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions. So you have one chemical reaction, you can put it together with another chemical reaction, put it together with another chemical reaction, put it together with another chemical reaction, put it together with the last chemical reaction, and then you can do all of these chemical reactions one after another to make a metabolic pathway, such as when the starting substrate molecule A is made into intermediate products B, and then C, and then D, and then E, and then the intermediate product E is made into the final product that we call the end product, and that would be molecule F. Any question about any of that? And this is a way that our cells make products. They will start with a molecule, use an enzyme to make an intermediate, use another enzyme to make another intermediate, etc., until the end product is made. So this is a metabolic pathway. Metabolic pathways are determined by enzymes, meaning you have a metabolic pathway running in our cells because of enzymes. And enzymes are encoded by genes. So everything comes down to the genetics. Any question about any of that? So if a cell wants to make end product F, and it has the starting molecule A, it will use a metabolic pathway to make the end product. You should know that molecule B is the product of this first reaction. Molecule C is the product of this reaction. And that's why we call these intermediate products, because they're intermediate molecules, but they are a product for their own reaction. Any question about any of that? The starting substrate will be called the substrate, but it can also be called the starting reactant. And you just need to know both of those terms. Sorry about that. The product, the end product, only has one word, end product. Anyways,
Professor, I do have a question.
uh-huh.
With these metabolic pathways, is it always just A through E for the molecules and then the end product F, or is there ever more than that? More
It de-
molecules than just A
yeah,
through E?
it depends on the metabolic pathway. Like for making sucrose, all you use are your starting molecules, and then in one step you make sucrose. So the starting molecules will be fructose and glucose. So that reaction only takes one step. Okay. And I can't give you an example that takes 5 steps. Is that 1, 2, 3, 4, 5 steps? But there are some. So it depends on the metabolic pathway, how many steps it takes. Does that answer your question?
Yeah, I was just wondering if it can go past F for it.
Yeah, it can go past F
Okay.
if more steps are needed.
Okay.
Okay. I can't even think of any that come to 5, so I certainly can't think of any that come to 6, but there are chemical reactions. And I could look in the next slide here. Here is all the metabolism that I want you to know. This is all the metabolism that they know that goes on within presumably a eukaryote cell, because I think that's aerobic respiration, which I guess does occur in prokaryotes. The point of the matter is there's a ton of metabolism going on in a chemical reaction. Well, here's one here, actually. In the Krebs cycle, which I think is shown right here, 1, 2, 3, 4, 5, 6, 7, about 8 steps in the Krebs cycle. And I'm not 100% certain it's the Krebs cycle, but it looks like. That was a joke when I said you had to know all of this chemical reaction. Nobody said, are you kidding? All right, let's talk a little bit more about chemical reactions, converting one molecule into another molecule. Chemical reactions occur by the collision theory. The collision theory states that chemical reactions may occur when atoms, ions, and molecules collide with each other. And if the two colliding components have enough energy, they will make the chemical reaction occur. So the activation energy is the energy needed to disrupt the electronic configurations of the starting substrates to make the product. So the activation energy. If you don't have enough energy in your collision, then you will not get the chemical reaction to proceed. But if you have enough energy, the activation energy, the two colliding products, and that could be atoms, ions, or molecules, will form the product. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes. That'll mean that the enzyme will be more likely to run into the reactants, and the enzyme will then make the products. So that'll speed up the reaction rate. Another way to speed up the reaction rate is you increase the temperature. If you increase the temperature, you increase the energy that the molecules have when they collide with each other. So you'll increase the reaction rate. And the pressure, you increase the pressure, it does the same thing, but it's a little more complicated, and I don't want to talk about pressure. So just increasing the pressure increases the reaction rate. Any question about any of that? I wonder if that's why food cooks quicker in a pressure cooker. Obviously, you can also get the temperature hotter, but it's also because of the pressure. So I would say yes, It's increasing the reaction rate. So let's look at the chemical reaction, looking at the activation energy. Enzymes reduce the activation energy of a chemical reaction. So when we have a chemical reaction, which we're graphing here, this is time on the x-axis, and this is the energy on the y-axis. So this initial substrates have higher energy than the products. So this reaction will tend to go because they have more energy than the products. So it's downhill. So this reaction is easier to proceed than a reaction that's uphill, meaning the reverse of that reaction, you could call it. Any question about any of that? All right.
I have a question. Is this an endothermic reaction?
Say again.
Is this an endothermic reaction?
Endothermic reaction. No, this is not an endothermic reaction because we're going to get more energy out of the reaction than we'll put in.
Okay.
So this would be an
Exothermic.
exothermic reaction.
Thank you.
Okay. So the activation energy without the enzyme is this energy hump here, meaning that to get these or this reactant to become products, they reactants have to get this amount of energy, which is called the activation energy without the enzyme, before the reactant will become the products. And that is the energy hump. If this was glucose, for example, we can make glucose into CO2 and water. And how you do that is you take a blowtorch to the glucose, and that will be adding the activation energy, the energy you need to convert glucose into CO2 and water. You would get more energy out. And if you don't know, if you were to burn glucose, it would give off an awful lot of energy and an awful lot of heat. In fact, some people who have made homemade bombs, glucose or sugar is actually one of the reactants they put in to making the bomb, because when you release that energy from the sugar, it gives off a lot of energy. Okay. And that would be this energy from here to there. We're not going to talk about that. We do need to talk about the activation energy with an enzyme. If on the other hand, you have an enzyme in the system, you don't have to add this amount of energy to get the reaction to proceed. And this isn't drawn correctly if this is glucose, because you do need to add a little bit of energy to the glucose to get CO2 and water, but you're not adding very much energy, because this would be the temperature of a blowtorch, which I don't know what that is, but it's very hot, something like 800 degrees Fahrenheit or maybe 1,000 degrees Fahrenheit. And don't ask me what it is Celsius, I don't know. It's an awful lot of energy. And the energy with the enzyme happens all the time in your cells, because your cells can convert glucose into CO2 and water. And obviously your cells are not going to be heated up to 1,000 degrees Fahrenheit. This reactant will get some energy, but it's not very much compared to adding a blowtorch to the glucose. And that amount of energy we call the activation energy with the enzyme. And you know it's not very high because your cells are at 37 degrees Celsius, which is 98.6 degrees Fahrenheit. Any question about any of that? All right. Enzymes are biological catalysts. They speed up chemical reactions. They can speed up a biochemical reaction 10 to the 8 to 10 to the 10 fold. You don't need to know these numbers, by the way. Enzymes are encoded by genes. So all the enzymes your cell has will be encoded by a gene. Enzymes are specific for a specific substrate. So generally speaking, an enzyme will perform one and only one single chemical reaction. This is a little bit of an oversimplification. As you know, we have some drugs which will mimic the reactant for the enzyme, but essentially an enzyme will work on one chemical reaction, one specific substrate. And yeah, it can bind to something that's very, very similar to that substrate. But like I said, that's a side note that we don't need to discuss in this class. So for this class, we're going to say that enzymes are specific for binding to one specific substrate. And the specificity of the enzyme is due to the enzyme's three-dimensional shape. So an enzyme acts on one specific substrate. You should know that enzymes are not used up or altered in the chemical reaction. So when the enzyme converts a substrate to the product, the enzyme is not used up, it's still there to convert more substrates to more product. And the enzyme at the end of the reaction is the same as the enzyme at the start of the reaction. So that we're saying is not altered in the reaction. And while the enzyme is converting the substrate into the product, you could argue that the enzyme is altered. We're not going to talk about that. We're going to look at only the enzyme before the reaction and the enzyme after the reaction, and then say the enzyme is not altered in the reaction. Any question about any of that? All right. So enzymes, how they convert a substrate to a product is the enzyme will actually physically bind to the substrate, forming an enzyme substrate complex. The enzyme orients the substrate and puts stress on the chemical bonds of that substrate to increase the probability of a chemical reaction occurring. And that is what's lowering the activation energy. That's how the enzyme lowers the activation energy, changing it from here to there. Any question about any of that? All right, you should know that most enzymes end in ACE, A-S-E, okay? We'll talk about some of these later in the term, ligase. Actually, that's the only one we're going to talk about. Catalase. A lot of enzymes end in ACE. Not all enzymes end in ACE, especially enzymes that were named before the scientists made this convention. One enzyme that we might talk about is renin. It curdles milk, and it's an enzyme very important in the baby's stomach to make the liquid milk stay in the stomach longer so it can be digested better. And that's about the only enzyme I know that doesn't end in ACE. You don't need to know all of these examples of enzymes ending in ACE. I need to say that enzymes do not always act in a vacuum I could probably say they never act in a vacuum. They have to have an environment around them for them to work. But some enzymes take a helper molecule before the enzyme can work. So some enzymes take either a coenzyme or a cofactor before the enzyme will work. And then when the helper is in there, the enzyme can now bind to the substrate where I haven't talked about the active site yet, but the enzyme actually can bind right here to the substrate. Okay. The difference between a cofactor and a coenzyme is a cofactor is mostly a metal. It's a cofactor is a single element, or you could say a single atom, usually metals. Excuse me. A coenzyme is a biological molecule like NAD. We'll talk about this in a little bit. NAD plus is a coenzyme. So the coenzyme is quite a bit bigger than a cofactor, which is usually one atom and usually a metal. And you don't need to know the holoenzyme name and the apoenzyme name. I'll never ask you about that. There are some important coenzymes. We're going to talk about two in aerobic respiration, so you need to know them. Two important coenzymes are NAD or NAD plus, and you want to know the name for NAD plus. It's nicotinamide adenine dinucleotide. It actually has a positive charge on it, so you should say NAD plus. It's a molecule we'll talk about in aerobic respiration, which is why you need to know it. A FAD, which is flavine adenine dinucleotide. It also is a molecule we're going to talk about in aerobic respiration. So you need to know this one. These other coenzymes, these two we're not going to talk about. You can find them in plants. Coenzyme A, we actually will talk about, but I'm not going to require you to know it as an important coenzyme. I think we talk about coenzyme A when we're talking about the Krebs cycle. I forgot that step, the preparatory step in the Krebs cycle. All right. So the mechanism that an enzyme acts is that an enzyme has an active site. Let me blow this up a little bit. An enzyme has an active site where that active site physically binds to the substrate. And when the enzyme binds to the substrate, we call it the enzyme substrate complex. And you can say that the enzyme binds to the substrate like the lock and the key model, where one key fits specifically to one lock. And then the enzyme puts stress on the bonds of the substrate, converting the substrate into the products. And then the product falls off of the enzyme, and then that enzyme can bind to another substrate. And that is lock and key model of an enzyme. Any question about any of that? Okay, well, there's also the hand in the glove model. And here we're looking at the hand in the glove model. It's a little more realistic. Here's our substrate. You can see it in 3D. There's our enzyme. You can see it in 3D. And here's when the enzyme and substrate bind to each other in the complex. And then that converts the substrate into the products, which isn't shown. And then this enzyme comes back to this, which can then bind to the substrate again. Okay? What do you notice about the picture here about the enzyme and the substrate? There's three things you should notice. So what's one? What's one thing you notice? Come on, one thing's very obvious.
The
It's even
substrate is on the
obvious.
outside versus the inside.
Say again?
The substrate is on the outside and the other one's on the inside.
That's true. The substrate does go on the inside, at least in this picture. That isn't always the case, where you could have an enzyme and the active site's right here, and then this binds right there. Obviously, this is not the correct picture for that, but the active site doesn't have to be on the inside. But in this case, it is. What's another thing you can notice about that? Something that's very obvious about the size of the molecules. Okay, we'll just sit here until somebody answers.
The molecules are very small.
The substrate is much smaller than the enzyme. The enzyme is much larger. What's another thing you notice about the difference here? Come on, come on.
I can't see the slide anymore.
I'll get it back. Where is that? There it is. All right. What do you notice about the enzyme between this picture and that picture? Is the enzyme acting like a lock or a key where it's not changing? Or I guess a lock doesn't change either. Or does the enzyme change?
The looks like the substrate is more closed on the right side.
Substrate of the enzyme changes its shape. You see that? And that's what we call it, the hand in the glove model. Let's see if I can blow this up. The third one is very difficult to see. If you look at the substrate right here and look at the substrate right there, you'll notice if you look really carefully that the substrate has changed its shape slightly. So the enzyme changes its shape, sort of like a glove, changes its shape to mold to the contours of the hand. Well, the enzyme is changing its shape when it's bound to the, not the enzyme, the substrate changes its shape when it binds to the enzyme. So this is more like the shapeshifter's hand molding to fit the glove, while the glove is molding to fit the shapeshifter's hand. And for those of you who ever watched Star Trek Deep Space Nine, this would be something like Odo putting his hand in the glove. Anyone got any questions about that?
Oh, go ahead.
Does substrate and enzyme both change their shapes too?
Yeah,
Okay.
that's what I'm saying. The enzyme clearly changes its shape. And if you look very, very closely, you have to look very closely. The substrate is flatter here than it is here. That's sort of pointed up and that's pointed down. This is flatter. The substrate when it's bound to the enzyme substrate complex. So both the enzyme and the substrate change their shape when they form the enzyme substrate complex. And that's why I said it's beyond the hand in the glove. It's the shapeshifter's hand changing shape to mold the glove while the glove is molding its shape to fit the shapeshifter's hand. All right, any question about any of that? Whoops. Didn't mean to go there yet. That's what I meant to do. All right, there are different factors that can influence enzyme activity. You'll go through this in your worksheet. So I'm just going to go through it quickly. Temperature, pH, and salinity, heavy metals, alcohol, and UV radiation can change an enzyme, causing the proteins, which are what the enzyme is made of, to denature. So you can change the shape of the enzyme by putting it at the wrong temperature, the wrong pH, the wrong salinity, having heavy metals around, having alcohol around, or exposing it to UV radiation. That can denature the protein, causing it to not work as well. And if you put it at extreme, like extreme pH, extreme salinity, a large number of heavy metals, a large amount of alcohol, you get it totally to denature, and then the enzyme won't function. You can also influence the enzyme activity by changing the substrate concentration. If you decrease the substrate concentration, you'll have the enzyme have less activity. If you increase the concentration up to a point, you'll increase the enzyme's activity. And that only goes to a point, and we'll talk about that later. Poisons can also influence an enzyme, and we're just going to say poisons generally stop the enzyme activity, like cyanide and fluoride. You have enough poison around, the enzymes won't work. And that's actually why cyanide is a poison to us, because it stops at least one of our enzymes. And that enzyme must be important for us to survive. I don't know what it is, so don't ask me. I don't even know what fluoride is too, but fluoride is a poison if we get too much of it. All right, various factors can influence the enzyme activity, and that includes an inhibitor. You can have two types of inhibition, competitive inhibition and non-competitive inhibition. Non-competitive inhibition is also called allosteric inhibition. And then lastly, we'll talk about feedback inhibition. All right, so this is an active functional enzyme. And as you can see, the protein is folded. For it to work, you can denature the enzyme. And then this is totally denatured. The enzyme will no longer work. One example of that would be when you, or that's not exactly an enzyme, but when you boil, not boil, but cook an egg, you have protein, and then when you heat it, you denature it, and that turns the egg white into a white part of the egg, meaning when you cook the egg. Temperature, all enzymes have an optimal temperature where they work best. This enzyme is working best around 37 degrees Celsius. So this might be an enzyme that works inside your body. And when you move away from that optimal temperature, the enzyme will have less and less activity. And when you move way away from that temperature, the enzyme will not work. Like when you go to boiling temperature, that's not actually boiling, but temperatures above 50 degrees, the enzyme will no longer work. It'll be totally denatured, and like it could be cooked. I'm not sure how hot 50 degrees is, but let's not get into that. And then when you freeze the enzyme, it won't work. And this enzyme loses its activity around refrigeration temperature. The same with pH. All enzymes have an optimal pH. This enzyme, the optimum is around pH 5. So what enzyme might this be? Where would this be in us? This enzyme which acts best around pH 5. Does anybody know?
It's acidic, right?
It's somewhat slightly acidic, yes.
Is it in your mouth? Is it like lipase?
It could be, yes, that would be a good one. This enzyme could be an enzyme that works in your mouth. I was thinking more of the skin. This enzyme probably works on your skin because the skin ranges from about pH 4 to pH 5.5. So my guess is this is an enzyme in the skin, but it could be an enzyme in your mouth because your mouth is somewhat acidic too. The trouble with the mouth is that the acidity in your mouth comes actually from the microbes in your mouth. So our own mouth without the microbes would not be at pH 5 or pH 5.5, whatever that is. And my point is, when you move away from that pH, the enzyme activity lessens. Oops, sorry. Until you go to extreme pH, like around pH 0 and pH 10, the enzyme has no activity. And if you were to ever to take egg white and then put it in vinegar or lemon juice, or put it in chlorine bleach and mix it, the egg white will actually turn white, meaning the egg, what do you call that? The runny egg white of the egg will then turn white, looking similar to egg white if you were to cook it. And I've done it in vinegar or lemon juice. I've never actually done it in bleach, but that would be putting it in an alkaline solution. Another factor that can influence the enzyme activity is if you increase the substrate concentration, you can increase the enzyme activity. So down here, if you have no substrate, you start increasing the substrate, you increase the activity of the enzyme up into a point. And then right here, when we increase the substrate concentration, you don't increase the enzyme activity any further. Why might that be?
Because the enzymes are already busy and
Yeah,
they do so much.
Yeah, the enzyme are already bound, all the enzyme are already bound to a substrate at this point. So if you increase the substrate higher, you can't increase the activity any further because all the enzymes are already bound. And that's why this graph initially increases rapidly, and then it starts to slow, and then it will plateau, and you can never increase it any faster. This similar graph would happen if you were to increase the amount of enzyme. And what you have in this point is when you're adding more enzyme is that all of the substrate is already bound to the enzyme, so you can't bind anymore. Let's see. All right, let's talk about inhibition. An inhibitor competes, stops an enzyme from acting. So an inhibitor stops an enzyme from acting. A competitive inhibitor competes for the active site with the substrate. So we can have a drug, which is a competitive inhibitor, and there's the enzyme, there's the substrate, they bind together in the active site. Instead, we can have the drug bind to the active site of the enzyme, and now the substrate can no longer bind to the enzyme. And that is how some drugs work. They are competitive inhibitors of enzymes. Any question about any of that? There are competitive inhibitors which are not drugs. I just know that there are some drugs which are competitive inhibitors. And don't ask me for an example. I don't remember one off the top of my head. But a competitive inhibitor binds to the enzyme's active site. A non-competitive inhibitor or an allosteric inhibitor binds to the enzyme at a site other than the active site. So there's the enzyme binding to the substrate. What we have is a non-competitive inhibitor or an allosteric inhibitor binding in the allosteric site That's outside of the active site. When the non-competitive inhibitor binds to the enzyme, it changes the folding pattern of the enzyme. And now the active site changes, and the active site can no longer bind to the substrate. And that's how non-competitive inhibitors act. Any question about any of that? All right. The last slide I want to talk about is feedback inhibition. It's a form of non-competitive allosteric inhibition. Feedback inhibition is where we have a chemical reaction occurring. So substrate is converted to intermediate A, which is converted by another enzyme to intermediate B, which is converted by another enzyme into the end product. Feedback inhibition is where the end product can actually go back and bind to one of the enzymes in the chemical reaction to make the end product. Usually, the end product binds to the first enzyme. Excuse me, I'm about to sneeze again. The reason why the end product binds to the first enzyme is that that end product acts as an allosteric inhibitor to shut down that enzyme. You shut down enzyme 1, you get no substrate made into intermediate A. There's no intermediate A, you cannot get intermediate B made. No intermediate B, you cannot get the end product made. So we effectively shut down all of this chemical reaction. I mean, we shut down all of this metabolic reaction. Why the end product tends to bind to the first enzyme is if you were to bind to a later enzyme, the cell would spend energy converting the substrate into at least intermediate A. And the cell doesn't want to spend energy that it doesn't need to spend because it's not going to convert the substrate into the end product. So most feedback inhibition occurring in a cell happens by the end product physically binding to the first enzyme and then inhibiting that first enzyme by feedback inhibition. And then we'll get no more end product made. Okay? Any questions about any of that?
Damn it.
You have a question? Ask.
Throw this on my charger in the room because I just unplugged this one. And then we need to look and see. There's soup.
Okay, I don't think they're talking to us. I need to shut that down. Let me
Question.
state that one other thing here, and that is, now let me think, what the heck was it?
I have a question, Dr. Volpe.
Uh-huh.
Where would this kind of inhibition be used?
No soup.
It's
What-
not irreversible. It's not irreversible. That's what I wanted to talk about further. And that is, if the cell needs more end product, that means there's not going to be end product around to bind to the enzyme. And the binding of the end product to the enzyme is an allosteric reaction, meaning every once in a while, the end product will fall off of the enzyme. When it falls off of the enzyme, if there's no end product around, this enzyme will then work, and then the chemical reaction will then proceed until we get enough end product made that it starts shutting down the chemical reaction. So this feedback inhibition has a way of shutting down the enzyme, but it also has a way of starting up the enzyme. When what am I saying? It has a way of starting up the metabolic reaction. Any questions there?
What would be an example of this?
I'm gonna have to say I don't have one off the top of my head. I will look into that and get back to you on it, okay?
Thank you.
Okay. All right, if there's no questions, I'm gonna end it here and I'll see you in the lab.
Can you put this PowerPoint on to available? And then could
This
you play
PowerPoint
maybe?
will not be available until I finish the lesson, but I will make the last PowerPoint available to you.
Okay, thank you.
Okay.